Hello, the title of the poster is Multi-omics Data Integration in the Cloud, using machine learning methods to predict statistically significant associations between clinical and molecular features between disparate breast cancer cohorts. The authors are Boris Aguilar, Kauterat-Villé from ISB-CGC, and George Acquaah-Mensah from the Massachusetts College of Pharmacy and Health Sciences. This work focuses on breast cancer, and it has been shown that relative to white women, Black women have a higher incidence rate prior to 40 years and a lower incidence rate after 50 years. Our aim is to use cloud-based multi-omics data analysis to identify novel associations between clinical and molecular features in breast cancer, and using machine learning methods to predict statistically significant associations between clinical and molecular features between the two cohorts, white and African American patients. We used the Google Cloud Platform for the analysis through the ISB-CGC, one of the National Cancer Institute's cloud resources. We used also TCGA dataset, which is stored or hosted by ISB-CGC in the form of BigQuery tables. We used the METhyl-mix package to analyze methylation data and gene expression data. We used Bicon to find biclusters, and we use machine learning tools, specifically deep learning and random forest, as implemented in Weka. The figure shows our pipeline. We analyzed two groups of patients, African American patients with stage 2 breast cancer with 50 years or younger, and white patients, stage 2 breast cancer, and also 50 years or younger. First, we use DNA methylation data and gene expression data to identify driver genes. Second, we use Bicon and gene expression data and protein-protein interaction networks to identify biclusters that are associated with the two cohorts. And finally, we used the driver genes to build predictors of the identified biclusters. For identifying biclusters, we used Bicon, a recently developed method that uses gene expression data and protein-protein interaction networks as input. This algorithm uses ant colony optimization, and also this algorithm can identify biclusters of patients and genes. METhyl-mix helped us to identify a list of genes, what we call driver genes, for which methylation levels and gene expression levels are correlated, and also that have methylation levels that are significantly different when compared to normal samples. This figure shows one of those genes, OTUB1, in which gene expression is negatively correlated with methylation. And in particular for this gene, there is a significant difference in gene expression when we compare white patients and African American patients. This gene is known to have an impact on immunosuppression in breast cancer. Bicon helped us to identify two biclusters, as you can see in this figure. And we found that there is a significant association between the cohorts, white and African American patients, and the bicluster membership. And the p-value of this association is 0.009. Our results shows that white patients are overrepresented in cluster 1, and African American patients are overrepresented in cluster 2. The biclusters are composed of genes that are differentially expressed between these two biclusters. And many of these genes are associated with severe patient outcomes. For example, low expression of ESR1 is associated with severe patient outcomes. And ESR1 has a low expression in bicluster 2 compared to bicluster 1. We used DM values to build predictors of biclusters. DM values are differences in methylation levels between cancer and normal samples, and they were computed using Methyl-mix. As you can see from the table, both methods, deep learning and random forest, can classify the bicluster 1 with a precision of 0.9 and bicluster 2 with precision of 0.7 for deep learning and 0.8 for random forest. We also use the DM values to build predictors of molecular subtype. And as you can see from this result, both methods can assign a basal subtype with a precision of 0.8. And in particular, deep learning can also predict LUM-A subtype with a precision of 0.8 and a recall of 0.8. In summary, we use gene expression, DNA methylation, protein-protein interaction networks to identify regulatory genes that are different in white patients relative to African American patients. Those include OTUB1 and FOXM1. We found 2 biclusters, and there was a significant association between cluster membership and patient cohorts. The trained algorithms were successful in predicting membership of the biclusters.